My research is on the sensory cues underlying competitive growth in the clown anemone fish. Many animal societies consist of cooperative breeding systems where members of the society forgo reproduction and help others to reproduce at some point in their lives. There are many species that partake in cooperative breeding, such as naked mole rats and meerkats. Selection might favor individuals that can adjust their growth to retain a lower rank or maintain a higher rank. In these cooperative breeding systems, access to breeding depends on an individual's position in a hierarchy, often which depends on an individual's size. Hutchard et al. Studied meerkats. They paired litter mates and fed the lighter of the pair half a hard-boiled egg twice per day for 3 months. These were identified as the challenger. They compared the growth of the challenger, the other litter mate, the challenged, and an unrelated control individual. You can see here their data, where the x-axis is the individual, either the control, the challenged, or the challenger, and the y-axis is growth, that individuals, when challenged, would increase their growth rate when a challenger's growth rate was experimentally increased. The increases of growth rate were associated with the increases of food intake. This shows that subordinate meerkats of both sexes would raise their own growth rate and food intake when same-sex rivals had their growth experimentally increased. This suggests that subordinates can track changes in the growth and size of potential competitors. While the exact cues that initiate competitive growth in meerkats is unknown, there are several other species that use different cues to assess size. Pygmy swordtails use aggressive encounters to assess size and recognize others. Male frogs use call frequency to evaluate the size of males in competition. Iberian rock lizards use tongue flicks to detect pheromones and assess the size of females. And snapping shrimp use the size of the open claw to assess body size of competition. So we ask the question, how do other animals assess size, and which sensory cues do they use when partaking in competitive growth? To answer this question, we used Amphiprion percula, or the clownfish, which live in social groups comprised of a breeding pair and 0 to 4 non-breeders. Within each group, there is a well-sized hierarchy. The female is the largest individual, the male is the second largest, and the non-breeders get progressively smaller. Clownfish are sequential hermaphrodites. If the female of the group dies, the male becomes the new female, and the largest non-breeder becomes the new male, which means that this would have been quite different. Clownfish also engage in competitive growth. Reed, a former student in the Boston lab, did a similar test to Hutchard, but in clownfish. Reed found that similar-sized individuals will increase the growth rate to outcompete rivals and attain the highest dominance rank. Knowing this, we ask the question, how do clownfish assess their own size, the size of others, and initiate competitive growth? We considered 4 hypotheses. They must have mechanosensory, pressure and/or touch, cues. Without mechanosensory cues, competitive growth will not occur. They must have auditory cues. Without auditory cues, competitive growth will not occur. They must have chemical cues. Without chemical cues, competitive growth will not occur. Or they must have visual cues. Without visual cues, competitive growth will not occur. The fish were measured from nose to the end of the spinal cord. You can actually see the spinal cord right here. And then size matched and separated into pairs plus a singleton. To test that mechanosensory cues were necessary to engage in competitive growth, no divider was used. This experiment allowed all 4 cues to be present: mechanosensory, auditory, chemosensory, and visual, as it would be impossible to block these 3 while allowing mechanosensory cues to remain. As you will see in the next few slides, all of the other tests had dividers in them to allow the presence of certain combinations of cues. This blue divider was placed in all of the singleton tanks as a sham control in order to ensure that both of the no cues blocked fish were offered equal amounts of food. A blue divider was inserted immediately prior to feeding and removed afterwards, as was performed in Reed et al. Also, as it was impossible to block auditory cues while allowing the other cues to remain, auditory cues were present in all tests. To test that auditory cues were necessary, an opaque solid divider was used. This divider was cemented in place to guarantee that no chemical cues passed through the water around the divider. The opacity prevented visual cues. To test that chemosensory cues were necessary, an opaque perforated divider was used. This had a mesh insert allowing water to flow from one side to the other. The opacity prevented the fish from seeing each other. To test that visual cues were necessary, a clear solid divider was used. This divider was cemented in place, preventing chemosensories from passing through, and the clarity allowed the fish to see each other. To test that chemosensory and visual cues were necessary, a clear perforated divider was used. The mesh insert allowed water to pass through the divider, and the fish could see each other through it. At the end of the month-long trial, the fish were euthanized and rated by size into ranks, P1 being the bigger of the pair, P2 the smaller of the pair, and S the singleton. So in the results, test 1 was the no cues blocked test. These are the fish that had access to mechanosensory, auditory, chemosensory, and visual cues. In both parts, the fish type is the x-axis, and fish growth is the y-axis. Growth is the difference between the final standard length and the initial standard length. The first part of the figure is comparing P1, P2, and S by growth. You can see that there's a significant difference between P1 and P2, P2 and S, and P1 and S. In other words, P1 grew faster than P2 and S, and P2 grew faster than S. When combining the P individuals, you can see the evidence of competitive growth as both paired individuals grew faster than the singleton. Test 2 was opaque solid. These are the fish that had access to auditory cues. Axes are the same. The first part of the figure is comparing P1, P2, and S by growth. You can see that there is a significant difference between P1 and P2, but not between P1 and S or P2 and S. When combining the P individuals, the paired fish did not grow more than the singletons, suggesting that the fish need more than auditory cues to initiate competitive growth. Test 3 was the opaque perforated. These are the fish that had access to auditory cues and chemical cues. Axes are the same as in the others. The first part of the figure is comparing P1, P2, and S by growth. You can see that there is no significant difference in growth between P1, P2, and S. When combining the P individuals, the paired fish did not grow more than the singletons, suggesting that the fish need more than auditory and chemosensory cues to initiate competitive growth. Test 4 was the clear solid divider. These are the fish that had access to auditory and visual cues. The axes are the same. The first part of the figure is comparing P1, P2, and S by growth. You can see that there is a significant difference between P1 and P2, but not between P1 and S or P2 and S. When combining the P individuals, the paired fish did not grow more than the singletons, suggesting that the fish need more than auditory and visual cues to initiate competitive growth. Test 5 was the clear perforated. These are the fish that had access to auditory, chemosensory, and visual cues. The axes are the same. The first part of the figure is comparing P1, P2, and S by growth. You can see that there is a significant difference between P1 and P2, but not between P1 and S or P2 and S. When combining the P individuals, the paired fish did not grow more than the singleton, suggesting the fish need more than auditory, chemosensory, and visual cues to initiate competitive growth. In order to provide further evidence of competitive growth, we performed a synthetic analysis. On the left, test type is the x-axis. So down here, we've got all 5 different tests. And growth of paired individuals is the y-axis. The figure is comparing the growth of all paired individuals per test type. The no cues blocked individuals, those with access to mechanosensory, auditory, chemosensory, and visual cues, grew faster than individuals of all other tests. The paired individuals from the other tests grew no differently from each other. On the right, the test type is the x-axis again, and the growth of singleton individuals is the y-axis. The figure is comparing the growth of singleton individuals per test type. All of the singletons grew similar amounts. In conclusion, we found that juvenile clownfish likely require the use of mechanosensory, pressure and/or touch cues to assess the size of conspecifics and engage in competitive growth. When auditory, chemosensory, or visual cues were available, but mechanosensory cues were not, competitive growth did not occur. When mechanosensory cues were present, one paired individual grew faster than the other paired individual, the paired individuals grew faster than the singleton, and the paired individuals that had access to mechanosensory cues grew faster than the paired individuals in any of the other tests. Our experiment cannot rule out the possibility that in some other element of interaction is permitted in test 1, the no cues blocks test. This could be swimming side by side using visual cues that might be stimulating competitive growth. Therefore, we recommend critically testing the mechanosensory cue hypothesis with a gentamicin knockout. Using the antibiotic to temporarily functionally block the neural masses of the lateral line in the inner ear from transmitting mechanosensory reception would allow the fish to have close proximity while negating the ability to detect mechanosensory cues. Endocrine mechanisms might underlie the phenotypic changes in the growth rate of clownfish. Some potential hormones could be arginine bacitracin, isotosin, sex steroids, or the growth hormone. Whole body ELISAs could be used to analyze the presence of hormones in homogenized samples. Some animals use auditory, chemosensory, and visual cues to assess size. And while less work has been published on the influence of mechanosensory cues in size assessment, we know that pygmy swordtails use fish bites, submissive fishes use quivering and head shaking behaviors, and the regular stimulation of the leg induces gregarious behaviors in locusts. We hypothesize that many taxa might be incorporating mechanosensory cues into their assessment of conspecifics. Ta-da. Hold on, let me stop recording.